understood, the distinct histological changes that characterise the remodeling of the vessel wall are being increasingly well defined. We know that infiltration of inflammatory cells is associated with pro-inflammatory mediator release. Indeed, there's a significant body of evidence to point towards an immune-mediated component. We know that there's release of proteolytic enzymes, and in the vast majority there's the presence of an intrarenal thrombus or ILT. And it's against this backdrop of a proteolytic inflammatory environment that the growth of blood vessel angiogenesis has been documented by a number of previous groups. However, the precise mechanisms and its significance to the disease are yet to be determined. More recently, the growth of lymphatic vessels or lymphangiogenesis has been highlighted in a number of conditions involving inflammation. These are therefore the various viruses. On the basis of our preliminary data, we hypothesised that both angio and lymphangiogenesis would be increased in AAA, and that the density of the microvessels seen would be with the extent of the inflammation, the thickness of the ILT, and with aneurysm diameter. Okay, so to test these hypotheses, we assembled cohorts of AAA tissue samples obtained from patients undergoing elective aneurysm repair. Alongside this, their preoperative CT scans were analysed to give a measure of ILT thickness and aneurysm diameter. In addition, samples of normal aorta were obtained from major match control under the GIFT project. Our primary mode analysis was immunogenesis. Okay, so this is a representative example from a AAA cohort. And the most striking observation initially is obviously the extent of inflammation. Inflammatory infiltration was present throughout all layers of the AAA wall within all of the aneurysm cohorts. There was also distinct positivity for both blood and lymphatic vessels, and these showed a close spatial relationship to each other and to the inflammatory infiltrate. So to look at this more closely, this is D240, which is a specific marker of lymphatic endothelium. And you can see on this example these distinct lymphatic vessels within an area of inflammation. All of the AAA samples were also characterised by high numbers of CD31 positive blood vessels. And in particular, these small, thin-walled, immature type vessels were abundant within the inflammatory area. We also looked at the expression profile of the matrix metalloproteins MMP2 and MMP9, as these enzymes are involved in both pathogenesis of AAA and lymphangiogenesis. The expression of both enzymes was increased in the AAA cohort, with MMP9 in particular being strongly positive in the inflammatory areas, where it appeared to localise again towards these vascular structures. So in order to quantitate these results, all slides for all markers were scored by 3 independent observers. In comparison, an area of high and low inflammation from the AAA samples and the normal tissues was all scored and subjected to the appropriate statistical test. Within the inflammatory AAA areas, the density of both blood and lymphatic vessels was significantly increased in comparison to non-inflammatory and control areas. And in the inflammatory areas, the degree of vascularization was also related to the extent of inflammation. This graph here shows the correlation between the number of D240 positive lymphatic vessels and the inflammatory count within the same field. CD31 positive blood vessels exhibited a similarly statistically significant trend. And using the CT analysis, we were able to calculate the thickness of the ILT and aneurysm diameter at 3 separate levels. And CD31 positive blood vascularity shows strong positive correlation with aneurysm diameter, which is obviously an established marker of disease progression. So in summary, both blood and lymphatic vessels were significantly increased in the AAA tissue and showed strong positive correlation with the extent of inflammation. It's possible that this increased vascularity can serve to augment the inflammatory response and contribute to disease progression. So the results from this study offer some new insights into the processes occurring in AAA. In particular, the discovery of lymphatic vessels, which to the best of our knowledge has not been demonstrated elsewhere. However, the conclusions are obviously based on the analysis of fixed tissues from within a small sample set. So therefore, it would be pertinent to move into an in vivo model to better understand the functional consequences of the vessels and the time course over which they occur. So one of these models would be the echo-efficient mouse, which when treated systemically then tends to develop suprarenal aneurysm. This is something we've been to look at. We've already had some positive staining for both blood and lymphatic vessels. And now moving on to compare this with normal mouse aorta and to look more closely at angio-lymphatic regulators. I'd like to support the sponsors of the project and say thank you to everyone who's contributed so far.
perusing results, why did your specific marker wasn't the line 1 marker? That's the first point. Have you compared your marker against that marker? Secondly, I have a problem with your controls, because I think you've used normal aorta, and it would be nice to use atheromatous aorta rather than normal aorta. And how do we not know that this is just an effect of a lot more inflammation? It has absolutely nothing to do with the process. So those 3 questions.
So to address the first one about live 1, we looked at both live 1 and D240 together in the initial period and found that they co-localized. But chose D240 purely practically because you don't need freshly cut sections, it's a more stable antibody, and in terms of just me doing it in the lab, it was easier to do it that way. And a number of studies have said that in sort of other systems. The second point was about the controls. A lot of the studies in this area suffer from the same problem, either there's such a paucity of any controls, or they use ones with concurrent aortic disease, which has been looked at previously and would be an angle that we could take it down.
Well, I think you certainly need to look at some atheromatous aorta by comparison.
Yeah.
And finally, is it not just an epiphenomenon?
Well, that's the kind of question, isn't it? I mean, I don't know. I think we've only got, like I said, we've only got a very small data set, and it's really difficult to comment either way. But it'd be nice to see which way this goes.
All right, Clever Clogs, what do you think? Epiphenomenon?
Yes.
Yeah, obviously do.
Can I just ask, were the pathologists who were looking at your slides aware of the central hypothesis of the study?
Were they aware of the central hypothesis?
Yeah, the central hypothesis of the study.
That's a good question. They knew we were looking at neovascularization in AAA, but they were blinded to whether the samples had come from either the AAA samples or controls.
Well, they weren't blinded because it's evident from the degrees of inflammation. That's the problem you see here, is that it's impossible to blind the samples, and we all know the best will in the world that the bias of what in terms of analysing subjectively immunohistochemistry is extremely high. I know from personal experience, even when you try to avoid it. Have you thought of doing an automated analysis?
That's something we talked about, but I mean, I'm sort of coming to this from a medical student standpoint, but in talking to my supervisors, I know that there's problems with the automated systems as well. I recognise the difference between the brown and the blue dots in particular, so nothing's perfect.
That comment about medical students playing with Joker, you know, so. Just to come to your controls, how many were on NSAIDs? And just in answer to that question there were 3 blinded observers, weren't there? So what was their correlation? How good were they correlate? Because that would be helpful actually in strengthening your...
Well, we did blind Goldman plots between the observers, and they came out in terms of variability, and hopefully they go into any potential paper, but just didn't have time to show them here. In terms of the NSAIDs,
I didn't think you would, that's why I asked the question. I think next time you should actually go back and look at that, because that's an important fact if you're going to get this paper published, which I'm sure you will. If I was refereeing, that's what I would say. Thank you very much. Excellent presentation.